In the last days of November 2014, a research vessel named Falkor sat almost motionless above the Mariana Trench, holding its position over the deepest wound in the surface of the Earth. The ship belonged to the Schmidt Ocean Institute, and the people on board were partway through a 30-day expedition called Hades, led by two scientists from the University of Hawaii, Jeff Drazen and Patty Fryer. Their plan was unusual for trench research. Instead of racing straight for the deepest point and planting a flag there, they were sampling the whole slope of the trench, from around 16,000 feet down to nearly 35,000, treating the abyss like a mountainside in reverse. To do it, they used machines called landers, which are about as unglamorous as deep-sea technology gets. A lander is a frame of metal and flotation with cameras, sensors, and a package of dead fish strapped to it. You drop it over the side. It falls, alone and unpowered, for hours. It sits in the dark, recording whatever comes to the bait. Then, on an acoustic command, it drops its ballast and floats back up to the world. One of those landers came back from a depth of about 8,145 meters. That's more than 26,700 feet of water, over 5 miles down. And when the team gathered in the ship's lab and played back what it had recorded, something pale slid into the pool of light around the bait. The first honest reaction from people who had spent their careers looking at deep-sea animals was confusion about what category of thing they were even seeing. It didn't look like an animal. It looked like debris, a scrap of something. One of the researchers on the expedition, a Hadal biologist named Alan Jameson, later described it as resembling a piece of wet tissue paper being dragged slowly through the water. It was whitish, translucent, so thin in places that the shadow of its own spine showed through its body. If you'd seen it drift past a pier, you'd have assumed a grocery bag had finally reached the end of its long journey. Then the debris steered. It turned against the current deliberately and held itself over the bait. Broad fins spread from its sides like soft wings, and a tail like an eel's began beating in long, patient waves. It had a face. It was hunting. And the moment that sank in, the depth reading stopped being a number and became a problem. Because this fragile, almost gelatinous creature, gliding around calmly at more than 26,000 feet, was the deepest living fish anyone had ever seen. Stop and sit with where this animal was. At the surface of the ocean, the air above you presses down with about 14.5 pounds on every square inch of your body. That load is called 1 atmosphere, and you never feel it, because you were built inside it. Descend into seawater, and you add roughly 1 more atmosphere for every 33 feet. That's the whole brutal arithmetic of the deep ocean. 33 feet down, the pressure has doubled. 100 feet down, the lungs of a breath-hold diver have been squeezed to a fraction of their surface volume. Where that snailfish was swimming, the water column overhead stacked up to more than 800 atmospheres. That's roughly 12,000 pounds pressing on every square inch. 12,000 pounds is 6 tons. Picture the weight of a large adult elephant, and now picture that entire weight concentrated onto an area the size of a postage stamp. And now picture that load repeated on every postage stamp of surface the animal has, from every direction, all the time. For its entire life, steel submarines with hulls engineered by naval architects have crumpled at pressures far gentler than that. The Falkor's team had lowered instruments in syntactic foam housings, rated by engineers precisely because ordinary equipment implodes. And here was a fish with skin like a soap bubble, a fish you could have injured by holding it too firmly, living in that pressure without any apparent effort at all. Nothing about it looked reinforced. Nothing about it looked armored. That was the detail that refused to fit, the one that made the first interpretation fall apart. Everyone who grows up on sea monster stories expects the deepest animal on Earth to look like a gargoyle, all fangs and plating and menace. The creatures that actually own the deepest water in the world are among the softest vertebrates alive. This is the question that will carry us all the way to the bottom. And I want to state it plainly, because the answer is one of the great underrated stories in biology. How does a living body survive pressure that destroys machines? And survive is the precise word here, because for anything made of flesh, resistance is off the table entirely. A body has to function inside the pressure, digest food, fire nerve impulses, pump blood, lay eggs, and build proteins under a load of 6 tons per square inch. The answer is a story about molecules, about fats behaving like butter in a refrigerator, about proteins being wrung out like sponges, and about a small, unglamorous chemical, the same one that gives fish their fishy smell, which may draw an absolute line across the ocean below which no fish can ever live. Scientists have actually calculated where that line should be. And the unsettling part, the part that will bring us back to this snailfish at the very end, is that the deepest fish ever recorded is swimming almost exactly on top of it. But you can't understand the bottom of the trench by starting at the bottom of the trench. Pressure is a ladder, and every rung changes the rules of chemistry a little more. Bodies that thrive at one depth are ruined at another. Solutions that work at 3,000 feet quietly fail at 10,000. So we're going to do what the ocean itself forces on every diving animal and every sinking scrap of food. We're going to go down slowly, one layer at a time, from the sunlit surface where pressure is a minor nuisance, through the depths that nearly killed the first human divers, past the deepest whales, into the black midwater where the real molecular warfare begins, and finally onto the floor of the Challenger Deep itself, where the fish stop, the crustaceans carry on. And the reason for that difference turns out to be written in the blood. The descent starts in the brightest, safest water in the sea. And the first thing to understand about pressure is why, for the first few hundred feet, it isn't really the water that's trying to kill you at all. Here's a fact that surprises almost everyone the first time they hear it. Water itself, under pressure, barely does anything. You can squeeze liquid water with the force of the entire ocean above the Challenger Deep, more than 1,000 atmospheres, and it'll only compress by around 5%. Liquids are already packed. Their molecules are shoulder to shoulder, and pushing on them harder mostly just makes them push back. And a living body, whether it belongs to a mackerel or a marine biologist, is overwhelmingly liquid. Muscle, blood, brain, skin, all of it is essentially water with ambitions. So when pressure climbs, the water inside a body and the water outside a body press on each other in balance, and the tissue itself is in no immediate danger of being crushed like a soda can. That's why the childhood image of the deep sea, the one where a diver who goes too far down is squashed flat like a cartoon character under a piano, has the physics backwards for most of the body. Flesh equalizes. If you were made of nothing except solid tissue and liquid, you could, in principle, tolerate astonishing depths before pressure touched you in any way you could feel. The problem is that almost nobody is made of nothing except solid tissue and liquid. The problem is gas. Gas is the traitor in the body. Unlike water, gas compresses beautifully, eagerly, in exact proportion to the pressure on it. Double the pressure, and a bubble of air shrinks to half its volume. Take it to 10 atmospheres, roughly 300 feet of seawater, and that bubble is down to 1/10 of its original size. Every airspace in a body is a structural weak point, a little pocket of surrender that collapses, while the flesh around it holds firm. And that mismatch, gas caving in while tissue stands still, is where the actual violence happens. Ask any scuba diver about their ears. The pain a diver feels 10 feet down in a swimming pool is exactly this story in miniature. The air-filled middle ear shrinking while the eardrum bows inward, tissue being dragged into a collapsing gas space. Divers equalize by forcing more air into that pocket. The deep sea, as we'll see, is full of animals that solved the same problem with a more radical strategy. They got rid of the pocket. To appreciate how expensive gas becomes with depth, you've got to look at the most elegant buoyancy device evolution ever built, and then watch it turn into a liability. That device is the swim bladder, and if you've ever kept goldfish or gone fishing on a lake in the summer, you've interacted with its owners. Most of the bony fish in the upper ocean carry a flexible bag of gas sitting in the body cavity, and its job is deceptively simple. Muscle and bone are denser than seawater. A fish made only of muscle and bone would sink and would have to burn energy swimming upward all day just to stay in place, like a man on an eternal treadmill. The swim bladder fixes that. By holding just the right volume of gas, the fish becomes weightless in the water, hovering without effort, spending its energy budget on hunting and escaping instead of on not sinking. It's a beautiful piece of accounting. Near the surface, it costs almost nothing and pays for itself every second. Now push that fish downward and watch the accounting rot. At 66 feet 3 atmospheres, its bladder has shrunk to a third of its volume, and the fish is suddenly heavy, sinking, forced to either swim constantly or refill the bag. Refilling means pulling gas out of its own blood and pumping it into the bladder against the outside pressure. And the deeper you go, the more savage that arithmetic becomes. A fish at 3,000 feet trying to inflate its bladder is doing something comparable to blowing up a party balloon, while 100 atmospheres try to blow it back down your throat. Fish actually evolved hardware for this. A gorgeous piece of plumbing called the rete mirabile, the wonderful net. A dense bundle of parallel capillaries that works as a countercurrent gas concentrator, packing oxygen into the bladder molecule by molecule against gradients that seem frankly unreasonable. Some deep-water fish operate these gas glands at depths of several thousand feet, and the existence of that machinery is about the best evidence I know that evolution has no pride. It'll build a cathedral of capillaries to keep a bag inflated if the energy math works out. But the math stops working. Below a certain depth, the cost of maintaining gas buoyancy eats too much of the food budget, and food, in the deep ocean, is the scarcest currency there is. So as you survey fish from the surface down into the permanent dark, you watch the swim bladder fade out of fashion like an old technology. Some deep-sea species keep the organ, but fill it with fat instead of gas, because fat holds its volume under pressure and never needs topping up. Others shrink it to a vestige. Many of the true deep-water residents, including the snailfish that opened our story, simply have none at all. Their buoyancy solution, and we'll come back to it, is to build the whole body out of materials that barely sink in the first place. Watery gel, soft reduced bone, tissue that hovers on its own. Where a surface fish is a dense machine slung under a balloon, a Hadal snailfish is closer to a water balloon with a nervous system, matched so nearly to the density of the sea around it that gravity almost loses interest. There's a lovely economic clarity to all of this. Gas buoyancy is like a mortgage with a variable interest rate pegged to depth. Cheap in the shallows, punishing in the midwater, impossible in the trenches, and the ocean's residents sort themselves along that curve exactly the way you'd predict, with the balloon owners crowding the sunlit floors of the building and the debt-free gel bodies living in the basement. For the animals that never leave one depth, that's the end of the gas story. But there's a whole category of creatures for whom the gas problem is far crueler, because they can't stay put. They live at the surface because they breathe air, and they feed in the deep because that's where the food is. Every working day of their lives is a commute straight down the pressure ladder and back. Air-breathing bodies packed with gas spaces, plunging voluntarily into water that treats every gas space as a demolition target. Their solutions are some of the most extreme engineering in the animal kingdom. And before we meet them, it's worth watching what happens when our own species, the only animal arrogant enough to attempt the same commute with hardware instead of anatomy, pushes its soft, air-filled body down the same ladder. Because humans have found their wall. We know exactly where it is. We know what it feels like to stand at it. and it's far, far shallower than where that snailfish lives. A human being on a single breath of air is a surprisingly capable diving animal. Our bodies carry an ancient package of reflexes, shared with seals and dolphins, that switches on the moment cold water touches the face. The heart slows, blood retreats from the limbs, and pools around the heart, lungs, and brain. The spleen contracts and releases a bonus dose of red blood cells. Trained freedivers ride that reflex to depths that sound invented. In 2007, the Austrian freediver Herbert Nitsch rode a weighted sled to a depth of 214 meters on one breath in the discipline called No Limits. That's around 700 feet. 700 feet down, his lungs, which started as the size of 2 footballs, had been compressed to something closer to the size of grapefruits, and blood plasma had shifted into the spaces of his chest to keep the collapsing airways from tearing. A trick borrowed, as we'll see, straight from the whales. One breath, a few minutes, and back. As a stunt, it's magnificent. As a way of living and working in the deep, it's useless. So humans brought gas down with them, and that's where the ocean started collecting payment. A diver breathing from a tank must breathe gas at the same pressure as the surrounding water, because lungs can't inflate against a pressure difference. And pressurized gas, once it's inside you, misbehaves. Nitrogen at a few atmospheres of pressure dissolves into the blood and the fatty tissue of the brain and starts acting like gin. Divers call it narcosis, or the rapture of the deep. And at 150 or 200 feet on ordinary air, experienced professionals have been known to offer their mouthpiece to a passing fish. Oxygen, the gas we die without, becomes actively toxic when its pressure climbs too high, capable of triggering convulsions with no warning. And any dissolved gas, on the way back up, threatens to come out of solution as bubbles in the joints, the spine, and the brain. The disease called the bends, which crippled and killed the caisson workers who built the Brooklyn Bridge more than a century before anyone understood the chemistry punishing them. Engineers and physiologists answered with a technique called saturation diving, one of the strangest occupations our species has ever invented. Saturation divers live for weeks inside pressurized steel chambers on ships or platforms, their bodies fully soaked with breathing gas at working pressure, commuting to the seafloor in a pressurized bell. They pay the decompression tax only once at the end, in a slow return to surface pressure that can take longer than a week. To sidestep nitrogen narcosis, they breathe helium mixed with oxygen. Helium is narcotically almost silent. It squeaks their voices into cartoons, wicks heat from their bodies so aggressively that the chambers must be kept tropical, and it lets working divers operate routinely at depths of 500 to 1,000 feet. The offshore oil industry runs on these people. It's a hidden profession, a few thousand humans on Earth who spend a month at a time living at 10 or 20 atmospheres, and I think it deserves to be far more famous than it is. But helium only removed the drunkenness. It uncovered something waiting underneath, and that's where the human story hits its wall. Push a helium-breathing diver past roughly 500 feet, compress them quickly, and the nervous system itself begins to fail. The hands develop a fine tremor that no act of will can stop. Reflexes turn jumpy. Divers report nausea, dizziness, sudden waves of sleep pressing down mid-task, thought turning slow and sticky. The condition is called high-pressure nervous syndrome, by syndrome, and the crucial thing to understand about it is that the cause is pressure itself, hydrostatic pressure, all by itself, reaching through the skull and interfering with the physical machinery of nerve cells, with the delicate proteins and membranes that carry every signal in the brain. At these depths, the human body has finally arrived where the deep-sea fish live all the time, at the place where pressure stops being a plumbing problem and becomes a molecular one. The shaking hands of a deep diver and the pressure-proof proteins of a snailfish are two answers to the very same physics, and only one of them works. The French commercial diving company Comex spent years probing exactly how deep a human nervous system could be pushed. Their experiments read like polar expeditions conducted vertically. In 1988, in the Mediterranean off Marseille, Comex divers in the Hydra 8 program performed real, working, open sea dives to 534 meters, more than 1,750 feet, breathing an exotic mixture that included hydrogen, because hydrogen, of all things, softened the tremors of the syndrome. That figure still stands among the deepest working dives in history. Then, in November 1992, in a pressure chamber in Marseille, a diver named Theo Mavrostamos was compressed over 13 days to a simulated depth of 701 meters, about 2,300 feet, held there breathing hydrogen, helium, and oxygen, and then decompressed for weeks. The entire experiment consumed 43 days of his life for a stay of roughly 2 hours at the record depth. Other divers in the same program, at slightly shallower, simulated depths, were already at the edge of what their bodies would tolerate. 3 decades later, nobody's gone deeper. Funding for such programs faded, true. Yet the deeper reason is that the physiology itself was announcing a boundary. Somewhere a few hundred meters past that mark, the consensus goes, the human nervous system under compression simply stops being reliably usable. Hold those two images side by side because the contrast is the whole point of this section of our descent. 2,300 feet reached once by one man after 13 days of careful compression at the cost of a 43-day confinement, with the tremor of high-pressure nervous syndrome hovering at the margins the entire time, and 26,700 feet occupied permanently by a fish that looks like it'd lose a fight with a strong cup of coffee, hunting and breeding and thriving under pressure more than 10 times greater than the pressure that stopped us. Whatever that fish knows, it does not know it in its armor, because it has none. It knows it in its molecules. Before we go down to meet those molecules, though, there's one more air breather worth following, because it makes our record attempts look almost comical, and it does so using nothing except anatomy. It weighs as much as 10 pickup trucks. It holds the deep diving record for all mammals in its family of tricks. And every single day it does voluntarily, dozens of times, what nearly killed the pioneers of deep diving. It solves the gas problem by embracing the collapse. A sperm whale preparing to dive does something that sounds like a mistake. It exhales. Every human instinct about deep water says the opposite. Take the biggest breath you can. Hoard the air. Ration it. The sperm whale, an animal that may spend an hour hunting squid thousands of feet down, heads for the deep with its lungs partly emptied. And the reason is a piece of engineering logic so clean it belongs in a textbook. Air, remember, is the traitor. A rigid chest full of gas at depth is a bomb waiting for permission. So the whale's body is built to let the traitor surrender early and completely. Its rib cage is jointed and flexible, where ours is a stiff cage, so the chest can fold inward without breaking. As the whale descends and pressure climbs, its lungs are allowed to collapse progressively. The air driven out of the delicate gas exchange tissue and into reinforced central airways, thick-walled pipes that hold their shape while the spongy parts of the lungs simply close down, like an empty accordion. Somewhere in the first few hundred meters, the lungs stop working as lungs at all. The whale is now, functionally, an animal without usable air inside it. And precisely because of that, the ocean has almost nothing left to crush. That raises the obvious question of what the whale is breathing down there. And the answer is that it's breathing its own muscles. Sperm whale muscle is loaded with myoglobin, an oxygen-storing protein packed in at concentrations roughly 10 times higher than in our muscle, enough to turn the meat almost black. The oxygen for the dive rides down chemically bound, dissolved into blood and tissue, in a form pressure can't compress and can't force into mischief. This, by the way, is also why the whale largely sidesteps the bends that terrorize human divers. With the lungs' gas exchange surface collapsed and sealed off early in the dive, there's very little nitrogen being pushed into the blood at high pressure in the first place. The whale did with anatomy what saturation divers do with steel chambers and decompression tables, and it did it a few tens of millions of years earlier. How deep does this take them? Sperm whales routinely work at depths of several thousand feet, and there's good evidence of dives beyond a mile. For a long time, they held the reputation as the deepest of all mammals. Then in 2014, a team led by Gregory Shore published the results of satellite tracking tags attached to Cuvier's beaked whales off the coast of Southern California, and the numbers embarrassed everything that came before. One tagged whale reached a recorded depth of 2,992 meters. That's about 9,800 feet, close to 2 miles of vertical water, a dive of well over 2 hours by an air-breathing mammal that must return to the surface for every molecule of oxygen it uses. The previous mammalian record, held by a southern elephant seal, fell by more than 1,000 meters in a single published paper. At the bottom of such a dive, the whale is under roughly 300 atmospheres, more pressure than any human has ever functioned in, and it spends its time there working hard. It's echolocating, chasing squid through absolute blackness, twisting, accelerating, and catching them. And yet, notice where even this astonishing animal stops. 2 miles down. The average depth of the ocean is a bit more than that. The beaked whale, the most extreme air-breathing diver we know of, is only just reaching the ocean's middle floor, and the trenches yawn another 4 miles below its deepest recorded point. The whale's solutions, the folding chest, the collapsing lung, the chemical oxygen, are all answers to the gas problem, and they're close to perfect. But the gas problem is only the first act of the pressure story. The whale visits the deep for an hour and leaves. Its proteins, its cell membranes, its nerves all remain surface equipment, built for 1 atmosphere, tolerating a temporary squeeze. An animal that lives in the deep permanently, that never surfaces, that builds its body and runs its chemistry down there from egg to death, faces an entirely different tier of the problem, and no amount of ribcage engineering addresses it. Because once the gas is gone, once a body is nothing except water, fat, and protein, pressure doesn't give up. It simply changes weapons. It stops attacking spaces and starts attacking substances. It goes after the 2 materials every living cell on Earth is built from: the oily film that wraps each cell and the folded protein machines that do each cell's work. This is the quiet war happening in every cubic inch of the deep ocean. And to see it, we've got to leave the divers and the whales behind, sink past the last blue glow of the surface world, and enter the layer where sunlight gives up. The residents of this place never see the surface. Their whole existence is conducted under a load that would make a saturation diver's hands shake. And the first thing pressure tries to take from them is something so humble, you've probably never once thought of it as life support technology. It tries to take the softness of their butter. Sunlight in the ocean dies slowly, and then all at once. In clear open water, the last usable light for photosynthesis fades out somewhere around 660 feet, and below that begins the layer oceanographers call the twilight zone, a belt of blue-black gloom stretching down to around 3,300 feet. This is where the permanent deep sea begins, and it's worth pausing on how much life we're talking about, because the twilight zone isn't a desert. By some estimates, it holds more fish, by sheer mass, than all the rest of the ocean combined, most of them small, silvery, and equipped with light-producing organs. Lanternfish alone, little animals you could hold 5 of in one hand, may be among the most abundant vertebrates on the planet. every one of those bodies, every night of its life, is running its chemistry at pressures between roughly 20 and 100 atmospheres. And at those pressures, the first molecular casualty is the cell membrane. Every cell you own is wrapped in a film of fat about 200,000 times thinner than this sentence is long, a double layer of oily molecules called lipids, arranged tail to tail like two crowds of people lying head out, feet together. The great secret of this membrane is that it works only because it's a liquid. It flows. Proteins float and drift through it like boats on a pond. Channels open and close by flexing within it. Nerve signals pass along it. Nutrients cross it. All of it depending on the film staying supple and mobile. Biologists literally describe the healthy state of a membrane with the phrase liquid crystalline, a substance ordered enough to hold together and fluid enough to move. Your life, moment to moment, is being conducted on the surface of a soap film that must never quite set. Now, what does pressure do to fats? You already know because you have a kitchen. Cooling a fat and pressurizing a fat do remarkably similar things to it. Both persuade its molecules to pack tighter, to snuggle into an orderly, rigid arrangement. Butter on a summer counter spreads like a dream. The same butter from the refrigerator tears the bread. Chemists call the rigid state a gel phase, and a membrane that drops into gel phase is a dead pond, frozen over. The boats stop moving. The channels jam. Nerves fall silent. Squeeze an ordinary surface water membrane down to deep sea pressures, especially in water that hovers a few degrees above freezing, and it stiffens toward exactly that fate. Pressure, in effect, refrigerates the deep twice, once with actual cold and once with compression. The fix that life discovered is the same one you rely on every time you pour salad dressing, and I find that oddly delightful. Olive oil stays liquid in the refrigerator, while butter turns to brick, and the difference lies in molecular geometry. The fat molecules in butter are mostly saturated, straight and tidy, and they stack against each other as neatly as pencils in a box. The fats in olive oil are unsaturated, meaning each carries kinks, permanent bends in the molecular tail, and bent molecules stack terribly. All those kinks jostle and hold each other apart, preserving looseness, preserving flow. Deep-sea organisms build their membranes out of olive oil logic. As species live deeper and colder, the proportion of unsaturated, kinked lipids in their membranes climbs, keeping the film fluid at pressures and temperatures that would set a surface creature's membranes solid. The strategy has a name: homeoviscous adaptation. That's a scientist's way of saying keep the thickness of the soup constant no matter what the world does. The name was coined in the 1970s by researchers studying how bacteria adjust their fats to temperature, and in the 1980s, the deep-sea microbiologists Edward DeLong and Aristides Ianos showed that pressure-loving bacteria hauled up from deep water do the same trick against pressure itself, retuning their membrane fats as the squeeze increases. The deeper the microbe's home, the kinkier its fats, as if every organism in the water column had read the same engineering memo. Fish obey the memo too. Analyses of deep-water fish tissue consistently find membranes rich in unsaturated lipids, tuned so that at the crushing cold pressure of their home depth, the membrane behaves about the way yours does at body temperature. Physiologists who study this sometimes describe the result as fine-tuning of the same dial across the whole depth of the ocean. A lanternfish membrane at its depth, a Hadal snailfish membrane at its depth, and your membrane at the surface are, in a functional sense, running at the same viscosity. Each is calibrated to feel identical from the inside while sitting in utterly different worlds. There's something quietly moving about that. Life settled on one target feeling for a membrane, and then invented 1,000 recipes for producing that feeling under 1,000 different loads. But notice the fine print, because it'll matter enormously at the bottom of our descent. A tuned membrane is tuned to its own depth, and that cuts in both directions. The deep specialist's loose, kinky, ultra-fluid membrane hauled up to surface pressure overshoots, turning too runny, too disordered, as leaky and sloppy as warm mayonnaise. This is one reason many true deep-sea microbes and animals can't survive being brought to the surface even when the temperature is kept perfectly cold. We tend to assume the deep is the hostile place, and the surface is the safe one. For most of the ocean's residents, our comfortable surface is the emergency. Most of the living volume of this planet is high-pressure habitat, and its residents are as fatally mismatched to our world as we are to theirs. Decompression, for them, is the disaster. Their butter melts. Membranes, though, were only pressure's first target, the soft opening move. Sink past the twilight zone, through 3,300 feet, and we enter the midnight zone, where no photon from the sun has ever arrived, and the pressure climbs past 100 atmospheres on its way toward 400. Down here, the attack goes deeper than the cell's wrapping. Down here, pressure reaches into the proteins themselves, the 10,000 kinds of folded molecular machines that carry out every action a body performs, and it begins, with perfect physical patience, to unfold them. Every thought you've ever had, every heartbeat, every twitch of every muscle, depends on proteins holding their shape. Now we get to watch the ocean try to take that shape away and discover the strange, smelly little molecule that takes its side. To understand what pressure does to a protein, you first have to appreciate what a protein is, because the word gets thrown around at the gym in a way that hides how strange these objects are. A protein begins its life as a chain, a long string of chemical beads called amino acids, assembled in an exact order dictated by a gene. And then, in a process that remains one of the deepest subjects in science, the chain folds. It collapses on itself in a fraction of a second, twisting and nesting into a precise three-dimensional shape. And that shape is everything. A protein is a machine whose function lives entirely in its geometry. Hemoglobin grabs oxygen because its folds form a pocket exactly shaped to hold it. Digestive enzymes slice food molecules because their folds form blades and clamps. The proteins in a nerve membrane pass electrical signals because their folds form gates that swing open at precisely the right voltage. Unfold any of them, let the chain relax back into loose spaghetti, and the machine is gone. The chemistry of being alive is, to a first approximation, the maintenance of tens of thousands of correct shapes. Here's the crucial physical detail, the one that hands pressure its weapon. A folded protein is full of emptiness. The folding is quick and imperfect by design, and the finished shape contains internal cavities, little pockets and seams where nothing sits, like the air gaps inside a hastily packed moving box. On top of that, a folded protein keeps much of its surface dry, tucking its water-repelling parts inward, away from the surrounding liquid. Both features share one property that matters to physics. They take up volume, and pressure is, at bottom, a force with a single obsession. It favors whatever arrangement of matter occupies less space. Squeeze a system hard enough, and it'll find its way toward compactness the way water finds its way downhill. An unfolded protein, counterintuitively, can occupy less total volume than a folded one, because when the chain opens up, water rushes into every cavity and packs tightly against every previously hidden surface. Water molecules snugged up against an unfolded chain pack more densely than they do in free liquid. So at great pressure, the physics starts whispering to every protein in a body the same seductive suggestion: unfold. Let the water in. Get smaller. At surface pressures, the whisper is far too weak to matter, since a folded protein is held together by an armory of internal bonds. But raise the load toward 400, 600, 800 atmospheres, and the whisper becomes a hand pressing on a latch. Delicate proteins begin to lose their grip on their own shape. Enzymes slow down and stiffen as their moving parts, which must flex and breathe to work, are pinned by the surrounding squeeze. Assemblies of multiple proteins, which hold hands across watery interfaces, get pried apart as water is forced into the joints between them. The wonderful phrase biochemists use is that pressure denatures proteins, the same verb applied to what a hot pan does to an egg. The clear part of an egg turns white in the pan because heat unfolds its proteins, and they tangle into a solid mess. Pressure can do the same job cold and silently. Squeeze egg white hard enough, thousands of atmospheres, and it'll set as though cooked without a single degree of heat. That last fact is worth a detour because it connects the bottom of the Mariana Trench to the refrigerated shelf of an American grocery store in a way I've always found wonderful. The premium juices and guacamole tubs labeled cold-pressed or high-pressure processed are preserved by exactly this physics. The food industry seals them in flexible packages, lowers them into water-filled steel vessels, and squeezes them to pressures around 4,000 to 6,000 atmospheres, several times the pressure at the deepest point of the ocean. Nothing is heated. The flavor survives. The microbes don't, because their proteins and membranes are torn apart by volume change, by the same molecular strategy the deep ocean uses on unprepared bodies, simply turned up beyond anything nature deploys. Somewhere in the supply chain of your breakfast, in other words, there are machines routinely producing conditions harsher than the Challenger Deep, precisely because pressure at that intensity is a sterilizing weapon. The deep sea's residents live partway up that same gradient of violence, permanently, and their existence means the weapon can be answered. Part of the answer, biologists discovered, is that deep sea life rewrites the machines themselves. Compare the same enzyme, say lactate dehydrogenase, a workhorse of muscle chemistry, across fish from different depths, and you find the deep water versions are subtly re-engineered, a few amino acid beads swapped here and there, producing a structure that's a little more compact, a little less cavity-ridden, a little less willing to let water force its joints. The physiologist George Samaro at Stanford, working with colleagues across decades, showed that these pressure-resistant protein variants appear again and again in unrelated deep-sea lineages, converging on the same structural fixes, the way distant cultures independently invent the arch. Evolution, given a few million years, will happily redesign a protein for depth. But redesigning every protein is a colossal project. A fish runs on tens of thousands of different protein machines, and pressure whispers to all of them at once. Rebuilding each one, mutation by mutation, without breaking its function, is the hard way to survive the deep. And the fossil record of genes tells us lineages that colonized the abyss did plenty of it. The elegant discovery, the one that reshaped this whole field, is that evolution also found a shortcut, a way to protect all the proteins at once without touching a single one of them. You do it by changing the water they live in. And the molecule that does it is one you've known your entire life without knowing its name. You've smelled it on every fishing dock, in every seafood market, on every wharf on Earth. The signature smell of the sea's animals, the fishiness of fish, is the calling card of the deep ocean's single most important anti-pressure technology. The molecule is called trimethylamine N-oxide, and since that's a mouthful even for chemists, everyone in the field just calls it TMAO. It's small, about as simple as organic molecules get, a single nitrogen atom wearing 3 little carbon caps and holding 1 oxygen. Fish tissue is full of it, and when a fish dies, bacteria get to work converting TMAO into its chemical cousin trimethylamine, which is volatile, drifts into the air, and lands in your nose as the unmistakable smell of fish. Fresh from the water, a fish barely smells at all. The fishy smell is TMAO's ghost. And for a long time, biologists filed TMAO away as a humble piece of housekeeping chemistry, one of several small molecules, collectively called osmolytes, that marine animals dissolve in their body fluids to balance the saltiness of the sea around them, keeping water from being sucked out of their tissue by osmosis. Useful, unglamorous, apparently boring. The person who noticed it was anything except boring is a comparative physiologist named Paul Yancey, a professor at Whitman College in Washington State, who had trained with George Somero and spent his career on exactly this question of how molecules cope with hostile environments. As Yancey and his colleagues measured TMAO levels across fish species, a pattern emerged with a cleanness that real biology almost never provides. The deeper a fish lives, the more TMAO it carries. A shallow water fish might run a modest baseline. A fish from the abyssal plain carries several times more. Hadal snailfish from the trenches carry the highest concentrations ever measured in fish. Plot TMAO concentration against the depth where each species lives, and the points fall along a strikingly straight rising line, as though every fish in the vertical mile after mile of ocean had been issued its dose according to the same simple formula. In biology, when a single chemical tracks a single physical variable that faithfully across dozens of species and multiple unrelated lineages, it's practically a signed confession. TMAO was doing something about depth. What it does, laboratory work confirmed, is defend protein shape. Chemists call molecules like this chemical chaperones, and the way TMAO chaperones is subtle and beautiful. Its real work happens out in the surrounding water. Dissolved TMAO changes the behavior of the water around it, strengthening the lattice of hydrogen bonds between water molecules, making the water, in a loose sense, more organized and more cohesive. Water that's less eager to be crammed into tight spaces. Remember how pressure attacks a protein by shoving water into its cavities and joints, paying for the crime with a reduction in total volume. TMAO raises the cost of that crime. Water that's busy holding hands with TMAO is water that resists being forced into a protein's interior. With enough TMAO dissolved in a cell, the unfolded, water-swollen state of a protein stops being the smaller option, and the physics that was whispering unfold falls silent. The protein stays folded because the water around it has been made unwilling to participate in the burglary. Osmolite researchers sometimes describe TMAO as pushing on proteins from the outside, counteracting the squeeze. And in the technical literature, molecules doing this job in deep-sea animals have earned a wonderful name. They're called piezolites, from the Greek word for pressure. Pressure pills effectively dissolved in the flesh of most of the vertebrate life in the deep ocean. Once you see this mechanism, scattered facts from all over marine biology snap into place. Deep sea fish taste and smell fishier than shallow fish, which cooks and fishmongers knew centuries before any chemist could say why. More depth, more TMAO, more ghost. Sharks and their relatives, which run their osmotic accounts on a different chemical, urea, face a problem because urea happens to be a protein destabilizer. And sure enough, their tissue carries TMAO in careful proportion to offset it. Two dissolved chemicals in a standing truce, with the deep water sharks and skates shifting the balance toward extra TMAO, exactly as the depth pattern predicts. Even human medicine has a strange echo of this chemistry. Our kidneys, whose inner tissue works in conditions of brutal chemical stress, concentrated salt, and urea, protect their own proteins with a cocktail of stabilizing osmolites. The same class of tricks, the same logic, running quietly inside your body right now. The deep ocean did the discovering first. Everything with proteins and hard circumstances eventually lands on the same solution. That's one of those facts that makes the living world feel less like a collection of species and more like one long argument with physics. I want to be careful, though, and say what the evidence does and doesn't establish, because this molecule is going to carry the ending of our whole story, and it deserves honest handling. The correlation between depth and TMAO is about as solid as marine biology gets, confirmed across many species, trenches, and research groups. The protein-stabilizing power of TMAO is likewise nailed down in laboratory experiments, where adding it to solutions measurably protects enzymes from pressure. What remains under active investigation is the full accounting, exactly how much of deep-sea survival this one molecule buys, how much is bought by the redesigned pressure-resistant proteins we met earlier, how much by the retuned membranes, and how the three systems trade off against each other in different lineages. The picture most researchers hold is of a layered defense, membranes tuned, proteins hardened, and the whole cellular interior bathed in stabilizing chemistry, with TMAO as the heaviest single contributor in bony fish. Layered defenses are what you build when a threat never ever stops pressing. And now the latter resumes, because TMAO has consequences, and one of them is a time bomb. The dose rises with depth. A fish that lives deeper needs more of it dissolved in every cell. Simple, linear, relentless. Ask the obvious next question, the one a child would ask. What happens when the dose can't go any higher? Hold that thought through the next few thousand feet of our descent, because the answer appears to be drawn on the map of the ocean itself at a specific depth, and the fish of the world stop almost exactly where it says they should. First, though, the descent itself. Below the midnight zone, past 13,000 feet, the seafloor opens out into the largest single environment on Earth. And before we can stand at the fish's final frontier, we've got to cross it. Because what lives there and what it eats sets the terms for everything in the trenches below. The abyssal plain is the default surface of planet Earth. We think of our world as a place of continents and mountains with some water in between, and the map lies to us daily on this point because the flat, sediment-covered seafloor lying between roughly 13,000 and 20,000 feet down covers more of the globe than every other kind of landscape combined. If you could drain the water away, the defining scenery of Earth would be an endless, gently rolling plain of soft gray clay, colder than a January night in Minnesota, dark beyond any cave, and pressed under 300 to 600 atmospheres. That's the ordinary condition of our planet's solid surface. The beaches and prairies and cities we consider normal are the thin, weird exception around the edges. Life on the plain is shaped by pressure mainly through hunger, and the connection is worth spelling out because it decides who can afford to live in the trenches later. Essentially, no food is produced in the deep sea itself, away from the special oases of hot vents and cold seeps. Everything eaten down there fell from above. Biologists call the falling material marine snow, a perpetual, thin drift of dead plankton, cast-off mucus, dust, and droppings, sinking for days and weeks, being nibbled at every level on the way down. By the time the snow settles onto the abyssal plain, the ocean above has taxed away all except a few % of the food value that left the surface. The deep sea floor is a banquet hall receiving crumbs through a mail slot, and that changes the economics of every adaptation we've discussed, because every defense costs energy. Synthesizing kinked lipids costs energy. Manufacturing TMAO by the gram costs energy. Running gas glands is out of the question. The abyss selects everywhere and always for bodies that spend almost nothing, move slowly, wait patiently, and treat every windfall as a festival. The windfalls are extraordinary when they come. When a whale dies at sea, 40 tons of concentrated food can drop out of the sky onto a plain that's been receiving crumbs for centuries, and the event, called a whale fall, becomes an ecosystem in itself. Sleeper sharks and hagfish strip the carcass over months. Crabs and snails and worms work the scraps for years, and then come the specialists that sound invented. In 2002, researchers using a submersible robot in Monterey Canyon, off California, found a gray whale skeleton furred with what looked like pink shag carpet, which turned out to be colonies of a previously unknown worm, later named Osedax, Latin for bone devourer. Osedax has no mouth and no gut. It grows root-like tissue into the bone and, with the help of internal bacteria, dissolves and digests the fats and protein locked inside the skeleton itself. A whale's bones, on the abyssal plain, are a multi-decade food deposit, and evolution produced a dedicated safe cracker for them. Nothing that falls is wasted. The mail slot economy makes waste unaffordable. Underneath all of this animal life, outnumbering it beyond any hope of comparison, are the microbes, and they force us to redraw our whole mental map of what pressure means to life. In the 1940s and 50s, the marine microbiologist Claude Zobel, working at the Scripps Institution of Oceanography, hauled up mud from deep water and showed that bacteria in it grew better in his laboratory when he put them back under pressure, an observation so contrary to expectation that the field took decades to fully absorb it. He called such organisms barophilic, pressure-loving. His scientific successor, Aristides Yayanos, whose name you heard earlier in the membrane story, built pressure-retaining samplers and, starting in the late 1970s, isolated and cultured deep-sea bacteria that went far beyond tolerating pressure. Some of them, recovered from the greatest depths, turned out to be obligately barophilic, meaning they require high pressure to grow at all. Bring them to the surface, hold them at 1 atmosphere, and they falter, their membranes gone sloppy, their protein machinery mistuned, until they die of what a physician, if microbes had physicians, would have to diagnose as decompression. Modern researchers, preferring Greek roots, call these organisms pisophiles, and they've been found thriving in samples from the deepest trench floors on Earth. Let the strangeness of that settle. For a piezophile from the abyssal plain, the pressure that would kill you within seconds is Tuesday morning. And the pleasant sunlit surface where you live, with its songbirds and its gentle breezes and its one comfortable atmosphere, is a lethal environment where its cells physically fall apart. Neither of you is the normal one in any absolute sense. Normal, in the end, simply means whatever a body's molecules were tuned for. And since the deep ocean is by volume the largest habitat this planet has, occupying vastly more living space than every forest, prairie, lake, and shoreline combined, a fair-minded alien cataloging Earth might reasonably conclude that high-pressure life is the planetary mainstream, and that we, the crushable creatures of the thin, bright margin, are the exotic extremophiles clinging to a dangerous edge. I find it a useful humiliation to keep in a back pocket. The plane, then, gives us the rules of the end game. Food nearly absent, cold absolute, pressure in the hundreds of atmospheres, answered by tuned membranes, hardened proteins, and rising doses of TMAO. Bodies built for patience and thrift. These rules hold across most of the floor of the world, and in a handful of places, the floor itself gives way. The plains are interrupted by scars where one plate of the Earth's crust bends beneath another and plunges toward the mantle, dragging the seafloor down with it into gashes so deep that the abyssal plain hangs above them, the way clouds hang above Denver. These are the Hadal Trenches, named for Hades, god of the underworld, and they're the final rungs of the pressure ladder. Below 20,000 feet, the ocean keeps another entire world, deeper beneath the abyssal plain than the plain is beneath the waves. It's time to go over the edge, and to return at last to the soft pink fish that started this story. Because the trenches are where its family stopped being an oddity and became a dynasty. Humanity first touched the bottom of its deepest trench with a weight on a rope. In 1875, the British survey ship HMS Challenger, 3 years into the around-the-world voyage that essentially invented the science of oceanography, paid out sounding line in the western Pacific, southwest of Guam, and kept paying it out long after the depths of any known ocean had been passed. The line finally found bottom at close to 5 miles, a figure so far beyond expectation that the officers ran the measurement again to be sure. One of the men responsible for such navigational work aboard was a young officer named Herbert Swire, and I mention him now because the ocean has a long memory, and his name is going to resurface in this story in the most unexpected place, attached to the very fish we've been following. The great gash the Challenger's rope had found came to bear the ship's name, the Mariana Trench, and its deepest chamber is called the Challenger Deep to this day, just under 36,000 feet at its most recent surveys, the lowest point on the solid surface of the Earth. The trenches as a group, a few dozen of them scattered around the Pacific Rim and beyond, hold what scientists call the Hadal Zone, everything below about 20,000 feet. Taken together, they cover a fraction of the seafloor so small it rounds to nothing, and yet they contain the entire deepest quarter of the ocean's depth range. They're also, and this surprised everyone who assumed the deepest place would be the emptiest inhabited. Every trench sampled has life in it, and the deepest trench floors positively swarm in places. But the community thins in a very particular way as you descend the trench walls, and the thinning is the plot of our final act. One by one, the great deep-sea groups drop out. The sharks are long gone, left behind above roughly 10,000 feet for reasons that appear to involve, at least in part, their urea-heavy chemistry and the energy cost of their bodies. Rat-tails, cusk eels, and the other professional fishes of the abyssal plain hold on down to around 20,000 feet, some a little beyond. Past that line, the fish community narrows to what is, effectively, a single family's franchise. The snailfish family, the Liparidae, is on its face the least likely candidate in the ocean for that honor. Snailfish are named for their vague resemblance to a snail's soft foot, and most of their more than 400 species are small, tadpole-shaped, scaleless creatures with loose, jelly-slicked skin found in tide pools, kelp forests, and cold shelf waters. Aquarists would describe the typical snailfish as unimpressive. If you lined up the ocean's fish families and asked a room of biologists to bet on which one would conquer the most hostile depths on the planet, the fortunes would have gone onto the armored, the toothy, the robust. The trenches gave the entire estate to the soft ones. In trench after trench around the Pacific, the deepest fish present is a snailfish, and each trench tends to host its own species, evolved in place, an archipelago of isolated dynasties in the deeps, like Darwin's finches scattered across islands that happen to be 5 miles underwater and upside down. The Mariana Trench's own dynasty became formally known to science in 2017, and the way it happened closes a loop a century and a half wide. Following the 2014 Falkor expedition, the team's traps eventually brought up dozens of specimens of the trench's dominant snailfish, the pinkish, large-headed species the cameras had watched swarming the bait. A group of researchers, including Mackenzie Geringer, then a doctoral student at the University of Hawaii, along with Thomas Lindley, Alan Jameson, Erica Goetz, and Jeff Drazen, described the new species from 37 specimens collected at depths between roughly 22,600 and 26,100 feet, which stood as the deepest well-documented capture of any vertebrate. They named it Pseudoliparus swirae, honoring Herbert Swire, the young officer from the ship that first measured the trench. The sounding line of 1875 and the fish of 2017, tied together in a single Latin name. Science does have a sense of poetry when it thinks nobody's watching. And the specimens themselves, examined on deck and in the laboratory, read like a manifesto for everything our descent has taught. No swim bladder anywhere in the family's deep species. The gas economy abandoned completely, exactly as the arithmetic demanded. In its place, buoyancy by construction. The body is loaded with a watery, gelatinous tissue, a subdermal layer of dilute jelly that's nearly the density of seawater, cheap to build in a food-starved world since it's mostly water, and utterly indifferent to pressure since it contains no gas and hardly differs from the sea it floats in. Gehringer's own research went on to show this gel is more than ballast, that it fills out the body contours and the tail in ways that can improve slow swimming, letting a fish built like dessert glide with surprising grace. The skin is scaleless and loose. The skeleton is reduced, lightly mineralized, more cartilage and suggestion than armor. The largest eggs are astonishing, up to nearly 4/10 of an inch across, among the biggest of any bony fish, each one provisioned like a lifeboat, a strategy for giving each of a few offspring a massive head start in an economy of crumbs. Nothing about the animal is hard. Its entire architectural philosophy is to present the pressure with nothing to break, to be so nearly made of ocean that the ocean can't find a grip on it, and then to let the invisible chemistry, the tuned membranes, the hardened enzymes, the heavy freight of TMAO, fight the real war molecule by molecule. Up close, the observers who've handled these fish keep reaching for the same startled vocabulary, and it echoes that first bewildered moment in the Falcor's lab. Fragile, translucent, almost missable. Jameson has made the point over and over in interviews and in his writing that the public image of Hadal life, the nightmare of fangs in the dark, has the situation essentially inverted. That terrifying-looking midwater creatures like the anglerfishes live far shallower, and the deepest livable floors of the planet belong to animals of almost absurd gentleness. Hollywood's never once gotten this right, and honestly, I sympathize with Hollywood because the truth performs terribly in a movie trailer. The most extreme vertebrate environment on Earth was won by the body plan of a soft, patient, faintly smiling tadpole. But a body plan is only the visible half of an organism. The other half is a text, 3 billion or so chemical letters of it. And in 2019, scientists finally opened the book and read what 5 miles of pressure does to a genome. The results turned out to be stranger and more moving than the anatomy, because written in the snailfish's genes is a record of everything its ancestors threw overboard on the way down. In 2019, a large team of researchers led by the evolutionary biologist Wen Wang published, in the journal Nature Ecology and Evolution, the first full genome of a Hadal snailfish, assembled from Mariana Trench specimens, alongside a detailed anatomical study. Reading a genome is the closest biology comes to reading an autobiography, because a genome records both what a lineage has built and what it's abandoned, and abandonment leaves scars. When a gene stops mattering to survival, mutations pile up in it unchecked, breaking it a little more each generation, until what remains is a recognizable corpse of a gene, a pseudogene, still sitting in the text, like a crossed-out paragraph. The snailfish genome is full of exactly such crossed-out paragraphs, and each one tells you what the deep decided this animal no longer needed. The eyes went first, or at least their finer functions did. The team found the genetic toolkit for vision gutted, with a large share of the light receptor genes lost or broken, in water that no photon of sunlight has reached for the entire history of the trench. Eyes are an expense with no revenue, and the Hadal snailfish retains only small eyes with limited function, a museum of a sense. The genes for skin pigmentation are damaged too. That's why the living fish is that unearthly translucent pink, the color of a body that stopped bothering to have a color. You're seeing, when you look at it, partly its blood and muscle, through unpainted glass. Nothing in the dark can see it. Paint costs protein. Then comes the discovery I find hardest to shake, the one that connects this genome to every anatomical oddity we've already met. The researchers found that a gene called BGLAP, which builds a protein known as osteocalcin, one of the key regulators of bone mineralization, carries a frame shift mutation, a typo that garbles the instructions partway through. The paper's authors linked this cautiously to the animal's soft, incompletely calcified skeleton, and to something stranger. The snailfish's skull, they reported, isn't fully closed. Where a surface fish seals its brain case into a rigid box of bone, the Hadal snailfish leaves gaps, and the researchers interpreted this as an adaptation with a brutal elegance. A sealed, rigid container is precisely the thing pressure destroys, and a skull is a container. Leave it partly open, keep it slightly soft, and the pressure inside the head and the pressure outside can stand in perfect balance with nothing rigid enough to fail. The same principle a submarine designer would recognize, solved by deleting the hull. Everything we said about the swim bladder, about presenting the ocean with nothing to break, turns out to be written all the way into the architecture of the skull. And the genome shows the exact typo where the hardness was surrendered. The additions are as eloquent as the losses. The genome carries expanded families of genes involved in fatty acid metabolism, consistent with the endless work of keeping membranes unsaturated and fluid, the olive oil program running at maximum. The team also reported extra copies of genes involved in transporting and processing the building blocks of cell membranes, and signals of strong activity in the chemistry that supports osmolytes, the TMAO economy we've already toured. Later work by other groups, sequencing more snailfish from other trenches, has kept filling in this picture, as Kiman si Fansha Testi Chekens. And one detail from the follow-up research deserves mention for sheer thematic perfection. Comparative studies reported changes affecting genes tied to the machinery of protein folding and repair, the cellular emergency services that refold damaged proteins, exactly the systems you'd reinforce if your entire environment were one long, slow attempt to unfold you. None of these individual genetic claims should be treated as beyond revision, since genome interpretation is a field that corrects itself constantly, and honest scientists flag many of these links as probable rather than proven. But the overall shape of the story is consistent across every study, and the shape is what matters. Vision gone, pigment gone, hard bone traded away, fat chemistry and protein defense reinforced. A ledger of sacrifices and investments, and every line of it points the same direction, downward. There's one more entry in the ledger, and it supplies a date. By comparing the Hadal snailfish genome with those of its shallow water relatives, the researchers estimated that this lineage split from its surface kin roughly 20 million years ago, with the push into the extreme depths happening more recently than that. 20 million years sounds like an eternity, and by human standards it is, but on the calendar of vertebrate evolution, it's strikingly quick for a transformation this total. Whales took about the same order of time to go from walking on legs to ruling the open sea. In a comparable window, a cold-water tadpole of a fish rebuilt its skeleton, discarded 2 senses worth of genes, re-engineered its fats, loaded its cells with protective chemistry, and took possession of an environment that kills steel. I hold the unfashionable opinion that this little pink fish is among the most impressive vertebrates that's ever existed, and I'm prepared to defend that opinion against all the tigers and eagles in the world, because tigers and eagles conquered places a body can at least visit. The snailfish conquered a place where physics itself campaigns against the existence of bodies. And yet, for all its completeness, the snailfish's victory has edges. Its dynasty rules from 20,000 feet down to somewhere past 27,000. The trenches keep going. The Challenger Deep bottoms out near 36,000 feet, and in that final unclaimed mile and a half, something changes. To understand what, we need to see the bottom itself, and the story of how human beings finally looked at it with their own eyes is one of the strangest episodes in the history of exploration, complete with a sighting that ichthyologists have been arguing about for more than 60 years. Because the first 2 men ever to reach the deepest point on Earth came back swearing they'd seen the one thing our entire molecular ladder says should never have been there: a fish. On the morning of January 23, 1960, in rough seas over the Mariana Trench, two men climbed down a conning tower into a steel sphere about 6.5 feet across and prepared to fall further than any human being had ever fallen. The craft was called the Trieste, and it amounted to a piece of applied stubbornness with a hull wrapped around it. Its designer, the Swiss physicist Auguste Piccard, had earlier in his career built a pressurized gondola and ridden a balloon higher into the stratosphere than anyone before him, then looked at the ocean and reasoned, with magnificent simplicity, that a balloon could be run in reverse. Instead of a bag of light gas lifting a cabin through the air, a float of gasoline, which is lighter than water and, crucially, is a liquid and therefore almost incompressible, would carry a heavy crude sphere through the sea, sinking under iron ballast and rising when the ballast was dropped. No cable to the surface, no propulsion worth mentioning, a deep-sea elevator with no building around it. The two men inside on that January morning were Auguste's son, Jacques Piccard, who'd inherited both the machine and the temperament, and a 28-year-old United States Navy lieutenant named Don Walsh. The descent took nearly 5 hours, and it was a slow tour of everything this story has described. Down through the last of the blue light in the first few hundred feet, through the twilight zone, where the water outside the single tiny window went black and stayed black, past the depth where high-pressure nervous syndrome ends the human working envelope, though the two men inside their sphere at 1 atmosphere felt nothing except the deepening cold, condensation slicking the steel around them, the hull ticking as it contracted, past the depth of the deepest whale, past the average floor of the ocean, and then far past it, into the trench, the sphere's walls now carrying loads no crude vehicle had ever asked of its hull. At around 30,000 feet, a sharp crack rang through the cabin like a gunshot. An outer acrylic window in the entrance tube had fractured under the strain. The sphere itself was sound, and the two men, having looked at each other and at their instruments, decided the crack was survivable and kept falling. It says something about the era and about those two particular nervous systems that the decision took only moments. Just after 1 in the afternoon, local time, the Trieste settled onto the floor of the Challenger Deep in a billowing cloud of pale sediment at a depth of roughly 35,800 feet. Nearly 7 miles of ocean stood between the two men and the sunlight. The pressure on the sphere was on the order of 16,000 pounds per square inch. That's roughly 8 tons on every square inch of steel, a total load across the hull measured in the hundreds of thousands of tons. Inside, two human beings at ordinary room pressure ate chocolate bars and looked out the window at a place no eyes had ever seen. They could stay only 20 minutes. And it was in those 20 minutes, in the settling dust of their arrival, at the most extreme address on the planet, that Jacques Piccard saw the thing that would outlive every other detail of the dive. Lying on the seabed, in the beam of their lights, Piccard reported a flat, pale animal, perhaps a foot long and half as wide, with 2 eyes visible on its upper surface. In his written account, he described it as a type of flatfish, resembling a sole, and he described watching it stir, move slowly out of the light, and disappear into the dark. To Piccard, the meaning was electric and obvious, and he said so for the rest of his life. If a fish, a true bony vertebrate, was living at the full bottom of the Challenger Deep, then the deepest hole in the ocean was fish country, and life's writ ran to the planet's absolute floor. The observation went around the world with the triumphant news of the dive itself. It entered textbooks. For a generation of readers, the fish at the bottom of the Challenger Deep was simply a fact, one of the things everyone knew. There was a problem, and the problem's grown slowly and steadily for 60 years. The Trieste carried no camera capable of documenting the encounter. No photograph of the animal exists, no film, nothing except the testimony of 2 exhausted, elated men peering through a small cone of light into a cloud of stirred sediment after 5 hours of descent in a place where no human perception had ever been calibrated. Every attempt since to confirm fish at anything close to that depth has failed. Cameras have been sent to the deepest trench floors again and again, baited, patient, recording for hours and days across multiple trenches and multiple decades. They've recorded swarming crustaceans in their thousands, sea cucumbers, drifting single-celled giants, and, in the deepest documented cases, the snailfish we've followed all through this story. Below about 27,500 feet, they've never once recorded a fish. Marine biologists who reviewed the sighting have pointed out that the trench floor hosts large pale sea cucumbers, some of them flattened, some of a foot or so in length that to a startled observer through a porthole could pass for a flat pale fish, and that certain features Piccard described, such as the pair of upward-facing eyes, may have been pattern completion by a human brain doing its best in an impossible moment. In 2012, two ichthyologists published a formal reassessment in a scientific journal bluntly subtitled Dispelling the Myth, laying out the case that the animal was, in all likelihood, a holothurian, a sea cucumber. Walsh himself, gracious to the end of his long life, acknowledged the uncertainty, while noting that they saw what they saw. And this is the point where I want to be extremely careful, both out of respect for two men of genuine and permanent courage, and because the episode teaches the single most valuable lesson in all of deep-sea science. The lesson of the Trieste flatfish is not that two heroes lied. The lesson is that eyewitness testimony, even from brave, brilliant, technically expert witnesses, is a fragile instrument, and the deep ocean is precisely the environment engineered to defeat it. One glimpse, one light beam, one stirred-up cloud of sediment, one brain primed by a lifetime of expecting fish to be everywhere water is. Science was right to be thrilled by the report, and science was right eventually to decline to build on it. What settles questions like this is repeatable, recorded, physical evidence, cameras that don't get excited, traps that hold what they catch, instruments that can be checked by rivals. By that standard, the deepest confirmed fish on Earth sits thousands of feet above where the Trieste landed, and the floor of the Challenger Deep remains, on all available evidence, a place without fish. Which forces the question our whole descent has been building toward. Why? The easy answer would be that scientists once again simply have too little data, that the fish are down there, and we've missed them. And the reason that easy answer deserves respect before we dismantle it is that the history of this field is one long record of the deep-making fools of confident experts. If you want to understand why today's researchers still refuse to say never about anything in the ocean, you've got to hear about the most spectacularly wrong idea in the history of marine science, an idea that was accepted, taught, and defended by serious people, right up until a broken telegraph cable came up from the deep wearing a coat of living animals. In the 1840s, the most influential marine naturalist in Britain was a brilliant, witty, endlessly energetic Manx scientist named Edward Forbes, and he was about to make the most instructive mistake in the history of ocean science. Forbes had sailed aboard a Royal Navy survey ship in the eastern Mediterranean, lowering dredges into the Aegean Sea and cataloging what came up, and he noticed a trend that was perfectly real. The deeper he dredged, the less he found. Shallow hauls came up, crowded with life. Deeper hauls came up sparse, and the trend line, extended with a ruler, hit zero. In 1843, he proposed what became known as the Azoic hypothesis, the claim that below about 300 fathoms, which is 1,800 feet, the ocean was a lifeless desert, too dark, too cold, and too pressurized for any living thing. It was a reasonable extrapolation from honest data, endorsed by a top scientist, and it fit everyone's intuition about the deep as a dead place. It was also spectacularly, planetarily wrong, about the great majority of the habitable volume of the Earth, and the evidence against it had already been sitting in specimen jars for decades. As far back as 1818, the Arctic explorer John Ross, sounding the seafloor in Baffin Bay, had hauled up a tangled, many-armed brittle star on a measured line of around 1,000 fathoms, and noted correctly that it must have been living where the line touched bottom. Naturalists in Norway, most famously the pastor-turned-zoologist Michael Sars and his son, were methodically cataloging hundreds of species dredged from well below the supposed line of death, including stalked sea lilies that looked like fossils come alive. The Azoic hypothesis absorbed these reports the way established ideas usually absorb inconvenient reports, which is to say it ignored them, explained them away as errors of measurement, contamination of the line, animals snagged on the way up. And then, in 1860, the counter-evidence arrived in a form nobody could wave off. A submarine telegraph cable in the Mediterranean, lying in water around 1,200 fathoms deep, more than 7,000 feet, failed and was hauled up for repair. It came up transformed. The recovered cable was crusted with living growth, corals and mollusks and worm tubes, animals that had settled on it, cemented themselves to it, and grown to maturity on the floor of a sea that the reigning theory declared uninhabitable. An organism cemented to a cable can't have been snagged in passing. It had lived down there for years at an address. The Azoic hypothesis died of embarrassment over the following decade, and the Challenger expedition finished the job in the 1870s by dredging living animals from every depth it could reach, including the floors of the deepest basins its lines could find. Life, it turned out, went all the way down. It had always gone all the way down. I tell that story for two reasons. The first is humility, because the pattern's repeated in softer forms ever since. Every generation of ocean scientists has drawn a line somewhere and said, below this surely things must finally thin out to nothing. And every generation has watched some instrument come back up and quietly retire the line. Nobody expected thriving communities in the trenches. They are there. Nobody expected the deepest trench floors to swarm. They swarm. Against that track record, a wise person keeps a standing discount on all claims of absence from the deep ocean. And working Hadal biologists will tell you, in nearly these words, that the ocean punishes the word never. But the second reason cuts the other way, and it's the reason the fishless floor of the Challenger Deep is a different kind of claim from the dead ocean of the 1840s. A stronger kind. Forbes had only a trend and an intuition. He extrapolated from thin data, with no mechanism, no physical reason why life should stop at 1,800 feet, and in fact, there was none. That's why his line evaporated on contact with better sampling. The modern claim about the deepest fish is built the opposite way up. It began with a mechanism. The TMAO arithmetic we're about to walk through, which predicted a specific stopping depth for a specific group of animals, for a specific chemical reason, before the best modern surveys were in. And then the surveys came in. Camera drop after camera drop, trench after trench, and the fish stopped where the chemistry said they should, while the crustaceans, which the same chemistry excuses from the limit, kept right on going to the absolute bottom. A prediction that lands like that has earned something no dead theory of the 1840s ever earned. It's earned the right to be taken seriously until a single swimming counterexample shows up. Science, at its best, holds both of these attitudes at once. The humility that remembers the telegraph cable and the confidence that respects a confirmed mechanism, and refuses to let either one silence the other. Before we walk through that final arithmetic, though, we owe the bottom itself a proper visit because since the Trieste's 20 minutes, human beings have gone back, with better machines and vastly better eyes. And what they found down there deserves to be seen on its own terms. The deepest place on Earth turns out to be stranger than both the dead void of a Victorian theory and the monster pit of the movie posters. a landscape at the pressure limit of biology, populated by the few kinds of creatures whose chemistry holds all the way down. The first return took 52 years, and the man who made it, alone, had spent the decades in between directing some of the most famous films ever made about the ocean. On March 26, 2012, the film director James Cameron folded himself into a pilot sphere 43 inches across, inside a lime green vertical torpedo of a submersible called Deep Sea Challenger, and dove alone to the bottom of the Challenger Deep. The machine had been built in near secrecy in Australia by Cameron and a team of engineers, and it embodied 50 years of lessons learned since the Trieste. Where the Trieste fell like a freight elevator, the Deep Sea Challenger descended like a dart, reaching in about 2.5 hours a depth the earlier craft needed 5 to touch. The reported bottom depth was close to 10,900 meters, in the neighborhood of 35,800 feet. The figures varying slightly between instruments and analyses, as measurements at those depths always do. And where Piccard and Walsh had one small window in 20 minutes, Cameron had lights, three-dimensional cameras, sampling gear, and hours. For the first time in human history, someone parked at the deepest place on Earth and simply looked around, carefully, at length, with recording equipment running. What he found, by his own account, was desolation of a very specific kind. He described the bottom as a vast, featureless, gelatinous plain, the color and apparent texture of the moon, stretching identically in every direction, without the slightest rise or landmark. He reported no monsters and nothing that read to the eye as a fish. What the dive and its associated science program did document was smaller, and for our story, far more meaningful. Amphipods, the little shrimp-like crustaceans we're about to spend serious time with, came readily to bait at full depth. Sea cucumbers were there. Sediment cores came up alive with microbes. And the expedition's science team ultimately reported dozens of species new to science, most of them microbial, some of them small invertebrates. The deepest place on the planet was confirmed to be inhabited, busily, so at the small end of the size spectrum, and empty precisely where the vertebrates should have been. Since then, the bottom of the Challenger Deep has gone from the least visited place on Earth to something almost like a destination. In 2019, the American investor and explorer Victor Vescovo, in a purpose-built titanium submersible called Limiting Factor, dove the Challenger Deep repeatedly within days, part of a program in which he piloted a submarine to the deepest point of all 5 oceans, with the Hadal scientist Alan Jameson, the same man who watched that first pale snailfish on the recordings in 2014, serving as chief scientist. Robotic vehicles from several nations have landed, sampled, and mapped. Crewed capsules have carried more people to the trench floor in the last several years than stood on it in the previous 6 decades combined. The accumulated picture from all of those visits is consistent, and it's worth stating cleanly, because it's the ground truth everything ahead stands on. The floor of the Challenger Deep is soft, pale, cold, and alive. Its visible citizens are crustaceans in remarkable numbers, sea cucumbers that trundle across the ooze, and, stranger than either, giant single-celled organisms called xenophyophories, amoeba-like beings the size of a spread hand that build crumbly sculpted homes out of sediment, some of the largest single cells known to exist anywhere. Life at full pressure, in other words, is present in abundance. It's just a very particular guest list, and every vertebrate on Earth appears to have been left off it. Of everything on that guest list, the amphipods are the headline act, and any camera lowered to the deepest trench floors makes the point within an hour. Amphipods are crustaceans, cousins of the sand fleas that hop around beach wrack, generally the size of a thumbnail up to, in the trenches, occasionally the size of a small lobster, a phenomenon called deep sea gigantism that scientists still argue about with genuine enjoyment. Drop bait at the bottom of the Challenger Deep, and amphipods arrive in minutes. First a few scouts, then dozens, then a boiling swarm of hundreds and thousands, stripping a chicken carcass or a mackerel to bone, with the efficiency of a piranha documentary. They're the sanitation department, the undertakers, and the opportunists of the Hadal world all at once, and they exist at every depth of every trench ever sampled, from the rims to the absolute floors. At depths where the last fish gave up more than a mile overhead, amphipods swarm in their thousands, apparently untroubled, eating, molting, breeding, and being eaten. In the Mariana Trench, the dominant deep species is a pale, hunchbacked little creature named Hirandelia gigas. And it's become quietly one of the most scientifically interesting animals in the ocean. Because whatever it's doing to live at 16,000 pounds per square inch, it's doing it better than any fish ever managed. And Hirandelia, studied up close, keeps producing surprises that sound like science fiction until you check the journals. Consider first what it eats when nothing falls from above. Trench floors collect not only carrion, but wood, coconut husks, leaves, whole trees flushed out of rivers by storms and typhoons, waterlogged until they sink, funneled by the trench walls down to the deepest line of the seafloor. Wood is almost pure cellulose, indigestible to nearly every animal on Earth without help from gut microbes. Japanese researchers studying Hirandelia gigas from the Challenger Deep reported that the animal produces its own cellulose-digesting enzyme, working at cold temperature and crushing pressure, an enzyme capable of turning sawdust into sugar in a laboratory dish. The deepest scavenger in the world can, in effect, eat lumber. In an economy of crumbs, at the final collection point of the entire ocean, evolution equipped the resident garbage crew to digest the garbage nobody else on the planet wants. And the enzyme that does it is itself a pressure-adapted protein, folded and functional at intensities that would seize up its shallow water equivalents. Every part of that sentence had to be discovered separately, and every part points the same way. This animal is finished work, a complete, polished, pressure-native design. The strangest Hirandelia discovery of all is the one that made headlines about armor, and it needs to be handled with the same care we gave the Trieste flatfish, because it's recent, striking, and rests on a smaller evidence base than the rest of our story. In April 2019, a team of Japanese researchers published a study in the journal PLOS ONE with a title that sounds like a comic book pitch. An aluminum shield, it announced, enables the amphipod Hirandelia gigas to inhabit deep-sea environments. The team had examined the exoskeletons of specimens collected from the deepest parts of the Mariana Trench, and their chemical analysis reported something no one had seen on an animal before. Coating the creature's shell, they found a layer containing aluminum in the form of a gel of aluminum hydroxide. Stranger still, they proposed the animal manufactures this coating itself through an improbable chain of chemistry. When Hirandelia digests its food, plant material among it, its gut chemistry produces a sugar acid, and the researchers showed in laboratory experiments that this acid, under conditions like those in the amphipod's gut, can leach aluminum ions out of swallowed seafloor sediment. Released at the shell's surface into seawater, which is mildly alkaline, the dissolved aluminum transforms into that hydroxide gel, wrapping the animal in a thin mineral overcoat. The team argued the coating does two jobs. It may buffer the shell mechanically against the extremes of its environment, and, perhaps more importantly, it seals the shell's calcium carbonate against the surrounding water, because the deepest ocean is chemically corrosive to carbonate. Below a boundary called the carbonate compensation depth, seawater dissolves calcium carbonate faster than organisms can lay it down, which means a trench-dwelling crustacean is effectively wearing bones made of a material its own habitat dissolves. A waterproof aluminum raincoat over the carbonate would answer a real and otherwise puzzling problem. I want to flag, honestly, how this claim should sit in your mind. It comes from one research group, in one published study on one species, And while the chemistry demonstrated in the laboratory is real and the elemental measurements on the shells are real, the interpretation that this is a functioning protective technology and a meaningful part of how the animal survives hasn't yet accumulated the pile of independent confirmations that surrounds, say, the TMAO story. Science journalism ran with the phrase, aluminum armor. It's irresistible, and I've just run with it too. The measured way to hold it is this. The deepest dwelling amphipod we know appears to carry an unusual aluminum-bearing coating that its own gut chemistry can plausibly produce. And if the interpretation holds up, it's one more example of the same theme our whole descent keeps sounding. An animal solving a physical assault with chemistry rather than with strength. If later work overturns it, the amphipod will remain no less astonishing, because the solid, many times replicated facts about Hirundellea, the swarming abundance at full depth, the pressure-adapted enzymes, the wood digestion are already more than enough to make it the undisputed champion animal of the deepest ocean. The important question is why? Why do crustaceans in general, and amphipods in particular, sail straight through the depth barrier that stops every fish? Pressure is identical for both. Cold is identical. Darkness and hunger are identical. Both run on proteins that pressure wants to unfold and membranes that pressure wants to stiffen. Yet the amphipods colonized the last mile and a half of the planet, and the fish didn't. And when a difference is that clean, that binary, across dozens of trenches, biology is practically begging you to find the variable that changed. The leading answer, and it's become one of the quietly beautiful ideas in marine science, has to do with salt, and with two fundamentally different ways of living inside an ocean of it. Seawater is considerably saltier than the blood of a bony fish. Fish blood runs at roughly a third of the ocean's saltiness, a fact usually explained as a fossil of history, an inheritance carried forward from ancestors whose chemistry was tuned in less salty waters. And that mismatch puts every marine bony fish in a permanent, expensive fight with physics. Water always moves across membranes from the less salty side toward the more salty side, which means the ocean is perpetually pulling water out of a fish's body, dehydrating it in the middle of the sea, like a castaway on a raft. Fish fight back by drinking seawater and burning energy to pump the excess salt back out through their gills, running desalination machinery every hour of their lives. Biologists call this strategy osmoregulation, holding your internal saltiness fixed and different from the world outside, and paying the bill forever. Crustaceans, along with most other marine invertebrates, never took up that fight. Their body fluids sit at essentially the same overall concentration as seawater. Nothing pulls water out of them, because inside and outside are already balanced. The strategy is called osmoconforming, and it amounts to agreeing with the ocean instead of arguing with it. Now bring back the TMAO story and watch the two strategies collide with depth in completely different ways. For a fish, TMAO is added into a body that's trying to stay far less concentrated than seawater. Every gram of protective chemical dissolved into its tissue raises its internal concentration, closing the gap between blood and ocean. The protection budget and the salt budget are the same budget. The deeper the fish, the more TMAO it must carry, the closer its internal fluids creep toward the concentration of the sea itself. And that gap, as we're about to see in detail, is finite. It can be spent down to zero, and at a calculable depth, it is. The amphipod's account works under different rules entirely. Already matched to seawater, it can swap the composition of its internal fluids, exchanging some salts for protective osmolytes, adjusting its chemical portfolio without any fixed ceiling imposed by osmosis, and Hadal crustaceans have indeed been found loaded with their own pressure-protecting chemicals, in some cases different molecules from the fish's TMAO, doing the same job of defending protein shape. Researchers studying them have reported levels that keep climbing in the deepest species, unbothered, past the point where the fish's arithmetic runs out of room. There's something almost fable-like in where this lands. The fish, the vertebrate, our own celebrated lineage, carried into the ocean an ancient refusal to be like the ocean, an insistence on maintaining its own internal country with its own rules. And that stubbornness is what finally caps its empire at 27,000 feet or so. The amphipod, an animal most people would step over on a beach without a glance, accepted the ocean's terms completely, and in exchange, the ocean let it have everything. Every trench, every floor, down to the last inch of the Challenger Deep. I don't think biology hands out morals, and I resist the urge to find one here. But as an accounting lesson, it's perfect. In the deep sea, sovereignty is a luxury good, and only the animals that gave it up could afford the bottom. That's the shape of the answer. Now we owe it the numbers, because this final piece of our story became real science, testable, falsifiable, publishable science, on the day somebody actually measured the fish at the bottom of the ladder and did the arithmetic in public. That happened in 2014, the same year the pale snailfish glided through the lights of the Falkors lander. And it started with 5 fish in a trap, hauled up from the Kermadec Trench off New Zealand. Before the arithmetic, one loose thread needs tying. Because it's been dangling since the divers of COMEX left the story with their trembling hands. High-pressure nervous syndrome is where our species felt pressure reach into its molecules, and we never finished asking the obvious question: what exactly was the pressure doing to those nervous systems, and how do the permanent residents of the deep, whose nerves work flawlessly at loads dozens of times greater, get away with it? The scientific trail here starts in one of the most unexpected places in all of physiology, with tadpoles in a pressure chamber. In experiments first run around the middle of the 20th century, researchers had anesthetized the animals in water dosed with a general anesthetic, the tadpoles drifting motionless, fully under. Then the researchers raised the hydrostatic pressure in the chamber to a couple of hundred atmospheres, and the tadpoles woke up. They began swimming around, and swimming around, anesthetized and active at once, as though the pressure had squeezed the sleep right out of them. Reduce the pressure, and they went back under. The phenomenon is called the pressure reversal of anesthesia. It's been reproduced in animals from newts to mice, and it's one of the great strange clues in neuroscience, because it says that pressure and anesthetics are, in some deep physical sense, opposing forces acting on the same machinery. Anesthetics, by the leading accounts, work by dissolving into and subtly loosening the nerve cells' membranes and channel proteins, expanding and disordering the delicate machinery until signals stop passing properly. Pressure does the reverse. It compresses, orders, and stiffens that same machinery. Push the two against each other, and they partially cancel. That's why a pressurized tadpole can shrug off a dose that would keep it under at the surface. And it's why high-pressure nervous syndrome is sometimes described, in a phrase I've always found chilling, as the opposite of anesthesia. The shaking hands of a diver at 2,000 feet are a nervous system being wound too tight, its membranes compressed toward rigidity, its ion channels, the folded protein gates that fire every impulse, being distorted by squeeze, its synapses releasing their chemical messengers out of balance. Interestingly, one practical countermeasure the diving physiologists found was to add a little nitrogen, a narcotic gas, a whiff of chemical anesthesia, back into the helium mixture, deliberately setting the two opposing distortions against each other inside the diver's own neurons. It helped. The record dives of the early 90s rode on exactly that kind of pharmacological tightrope. Now look at the snailfish's nervous system with those experiments in mind. Every mechanism our descent has assembled applies with special force in nerves, because nerves are the most pressure-exposed machinery a vertebrate owns. Their function depends entirely on membrane fluidity, the very property pressure attacks first, and on the split-second shape changes of channel proteins. The very motions pressure pins down. A Hadal fish's nerves work at 800 atmospheres for the same layered reasons the rest of it works. Membranes tune to extraordinary looseness at surface pressure, arriving at correct fluidity only under load. Channel proteins whose structures, comparative studies suggest, carry the same style of pressure. Resistant retuning found in the deep enzymes, and the whole apparatus bathed in TMAO-rich fluid that defends every protein's folded shape. A human nervous system at depth is surface equipment being crushed toward malfunction. A snailfish nervous system at the surface would be, in the most literal sense, equipment too loose to function properly, overfluid, misgated, out of tune. Each one is calibrated to its own atmosphere count, and the calibration goes all the way down into the physics of the synapse. When the pale fish in the lander's lights turned against the current and steered, calm and precise, 5 miles down, every flick of that steering ran on impulses whose underlying chemistry had been rebuilt, molecule by molecule, to fire correctly in conditions that reduce our own neurons to static. There's a final twist in the nerve story that I find almost unbearably elegant. Recall that the diver's syndrome was tamed, partially, by mixing an anesthetic into the breathing gas, balancing squeeze with loosening. Evolution, it appears, may have discovered a version of the same trick. Some researchers who study deep-sea adaptation have pointed out that heavily unsaturated extra-fluid membranes and certain osmolytes can act functionally like a permanent, precisely dosed counterweight to pressure, a standing chemical loosening built into the tissue, holding the nervous system at its correct operating point the way the nitrogen in the diver's mix held theirs, except tuned over millions of years instead of improvised over weeks in a chamber. The comparison should be taken as an illustration rather than a strict identity, since the molecular details differ, but as a way of seeing the unity of the problem, it's hard to beat. A French diver in 1992 and a snailfish in the Mariana Trench were solving the same equation, one with a gas blender and 13 days of compression, one with 20 million years of natural selection. The fish's answer is better. It'd be strange if it were otherwise, given the difference in development time, and I say that with full admiration for the gas blenders, so the ledger of survival is complete, from skin to synapse. Gas spaces deleted. Bodies matched to the density of the sea. Membranes kinked into permanent fluidity. Proteins recarved into compact cavity-poor, pressure-proof forms. The cellular sea inside every cell dosed with a protective chemistry that thickens with depth on a fixed schedule. Nerves retuned to fire under load. Skulls left open, so nothing rigid can fail. Eggs the size of lifeboats. Patience in place of speed. Thrift in place of power. It's a complete design philosophy, coherent from the scale of the whole animal down to the scale of individual water molecules being organized around a single dissolved chaperone. And precisely because the design is so complete, so obviously capable of iterating further, the fact that it stops, cleanly and universally, at a particular line on the depth chart of the world ocean, demands an explanation of the same quality. That explanation exists. It's quantitative, it's testable, it's survived a decade of attempts to break it, and it fits on the back of an envelope. Here it is. In 2011, a deep trap set in the Kermadec Trench, northeast of New Zealand, came up from around 23,000 feet carrying 5 snailfish of the species Notalaparis kermitacensis, that trench's own resident dynasty, cousins of our Mariana fish. 5 specimens doesn't sound like much, and for most of biology, it'd be a footnote. For Paul Yancey, it was the missing rung of a ladder he'd been climbing for 30 years because these were the deepest fish anyone had ever gotten fresh into a laboratory. And that meant, for the first time, the TMAO measurement could be made at the very bottom of the fish's world. Yancey, together with Mackenzie Geringer, Jeff Drazen, and colleagues, ran the tissue chemistry, and in 2014, they published the result in the Proceedings of the National Academy of Sciences, under a title that's a model of scientific understatement. Marine fish may be biochemically constrained from inhabiting the deepest ocean depths. The measurements landed exactly on the long, straight line. The Kermadex snailfish carried the highest TMAO concentrations ever recorded in a fish, several times the load of a shallow water species, precisely what the depth trend predicted for an animal living around 23,000 feet. And with the line now anchored by real data at its deep end, the team did the simple, devastating thing. They extended it. The logic runs like this, and it needs nothing beyond what our whole descent has already established. TMAO dissolved in a fish's fluids does two things at once, inseparably. It protects proteins, and it raises the concentration of the fish's internal fluids. A fish's blood starts far less concentrated than seawater. As depth increases and the TMAO dose climbs along its straight line, the fish's internal concentration creeps upward, closing the gap with the ocean outside. Follow the line downward through the water column, and you can calculate the exact depth at which the gap closes completely, where a fish's fluids would have to match full-strength seawater just to carry enough TMAO to keep its proteins folded. The number that came out of the arithmetic was a depth of roughly 8,200 meters. In feet, just under 27,000. The paper gave the window around it as roughly 8,000 to 8,500 meters, allowing for the uncertainties. That's about 26,200 to 27,900 feet. Below that line, according to the hypothesis, a fish faces an impossible choice. Carry less TMAO than the pressure demands, and its proteins begin, slowly and fatally, to lose their grip on their shapes. Carry the full dose, and its internal fluids pass the concentration of seawater, at which point the ancient osmotic arrangement of every bony fish flips into reverse. Water would begin flowing into the fish instead of out of it, and the entire suite of machinery a marine fish uses to keep itself from dehydrating, the drinking of seawater, the salt-pumping gills, all of it would be running backwards against a problem it was never built to face, a swelling body in need of bailing out rather than topping up. Some animals, for the record, do live hyperosmotic to seawater and manage the influx. Fish, in 500 million years, have never rebuilt themselves to do so, and the hypothesis says the trench floors are asking them to do exactly that, as the cover charge for the last mile and a half. Now put the predicted line on the map of the world ocean and lower the cameras. That's the part that, when I first understood it, raised the hair on my arms. In 2017, in the Mariana Trench, a camera system recorded snailfish swimming at 8,178 meters, about 26,830 feet, deeper than any fish ever confirmed before and just above the predicted line. Then, on the 15th of August, 2022, in the Izu Ogasawara Trench, south of Japan, during an expedition led by Alan Jameson, working with scientists from the Tokyo University of Marine Science and Technology aboard the vessel Pressure Drop, a baited camera at 8,336 meters, 27,349 feet, recorded a small, almost transparent juvenile snailfish drifting calmly past the bait. Published in 2023, it stands today as the deepest fish ever documented by anyone, anywhere. On the same expedition, traps at 8,022 meters caught 2 snailfish in hand. The deepest physical capture of a fish in history set those numbers against the prediction. The line said fish should end somewhere between 8,000 and 8,500 meters. The deepest fish ever seen was swimming at 8,336. That animal was inside the predicted terminal window, a fish living within roughly 1 or 2% of the calculated absolute limit for its entire evolutionary lineage. Jameson himself has said publicly that he expects any future record to move by tens of meters and no more because in trench after trench, across more than a decade of systematic camera work, the pattern repeats without a single exception. Snailfish to the vicinity of the line. Below the line, amphipods in their swarming thousands, sea cucumbers, xenophyophores, microbes, an entire functioning ecosystem, and no fish, ever, anywhere. The deepest quarter of the ocean's depth range, everything from about 27,500 feet down to 36,000, appears to be, for vertebrates, a country with a chemical border no member of our lineage has ever crossed. Understand what an odd kind of triumph this is. The ocean's fish are stopped, on the best current evidence, by a molecule. The same molecule that saves them. TMAO carried them down through 5 miles of increasing crush, gram by gram, rung by rung, the single most effective anti-pressure technology vertebrate life ever found, and the very property that makes it work. Its presence as a dissolved substance in the body's water is what writes the invoice that finally can't be paid. The barrier is osmotic arithmetic, a consequence of decisions locked into the vertebrate body plan hundreds of millions of years ago in ancestors who never saw a trench. A predator guarding the last mile and a half would have been a simpler story. So would a temperature wall or a shortage of food. And the amphipods down there are demonstrably well fed. If you want a one-sentence summary of everything this descent has taught, it might be this. The animals who went deepest are the softest things in the sea, carried down, rung by rung, by their own chemistry, and stopped, at last, by the bill for that same chemistry coming due. And yet, notice the words the scientists themselves keep choosing may be constrained. The hypothesis, the predicted window. That care is deliberate, and it marks the honest edge of this whole magnificent story because the TMAO limit, for all its confirmations, remains a hypothesis about a place we've barely begun to search, and the people who built it are the first to list the ways it could still be wrong. Here, then, is the state of the argument at the honest frontier, and I want to lay out the doubts with the same respect we gave the evidence. Because the doubts are serious. They're held by serious people, and some of them are held by the very researchers whose work built the hypothesis. The first doubt is about sampling, and it's the ghost of Edward Forbes rattling its chains. The Hadal Zone's been surveyed by a handful of research groups, deploying baited cameras and traps at a few hundred sites across trenches whose combined floor area rivals the size of a continent's coastal plains. Baited cameras see animals that come to bait. A fish that ignored carrion, a specialist feeding on something else entirely, hugging a trench wall, rare, sparse, or shy, could in principle be down there below the line right now, unphotographed, and the field is too young and the place too vast for anyone honest to claim the search is complete. The counterweight is that the search, where it's looked, has been strikingly consistent, with snailfish appearing reliably to the vicinity of the predicted line and never once past it, in any trench, on any continent side of any ocean. While the same cameras at the same deeper stations record abundant life of every other kind. Absence with that much structure starts to carry information. It's the difference between not finding your keys anywhere in the house and finding them in every room except one, always, over a decade of looking. The second doubt is about time. The trenches are, geologically speaking, restless and comparatively young environments, and the snailfish dynasties, remember, took possession within the last several million years, with some estimates for particular trench populations far more recent. Perhaps this argument runs, the fish simply haven't had time yet. Perhaps the osmotic wall is real but temporary, and given another 20 million years, Some snailfish lineage will do what the hypothesis says would be required: rebuild its water-handling machinery, cross into hyperosmotic territory, and colonize the final floors. Perhaps some lineage somewhere is partway through that transition today. The TMAO limit describes fish as they're currently built. Evolution's whole business is renovating what is currently built. And 500 million years of vertebrate history is a long record of walls that turned out to have doors. The third doubt is about the arithmetic itself. The linear TMAO trend is drawn through the species we've measured, and the deepest measured point sits well above the line's end. The extrapolation across that last unmeasured stretch assumes the trend keeps its slope, that no additional chemistry appears, that no partial substitute for TMAO exists that protects proteins without the same osmotic cost. Deep-sea biology's been surprised by unadvertised chemistry before, and the deepest snailfish have already shown other tools in their kit: the hardened enzymes, the reinforced protein repair systems, whose combined sealing nobody's calculated from first principles. The predictions behaved beautifully so far. So did a great many predictions in the history of this field, right up until an instrument came back up holding a correction. So this is where I get to say the thing a documentary rarely gets to say, which is that nobody knows. And that the not knowing is specific, structured, and gorgeous. What's known is this: fish, carried by a molecular chaperone whose dose rises in a straight line with depth, occupy the ocean down to about 8,336 measured meters, roughly 27,300 feet, and have never once been recorded below that, while a calculation grounded in well-tested chemistry says their body plan should fail somewhere in almost exactly that band, because the protective dose, which would push their internal fluids past the concentration of the sea itself. What's open is whether that calculated wall is truly absolute, for all fish, for all time, or whether it's a description of an evolutionary moment, a border that patience, or a chemical trick not yet discovered, or simple further searching will eventually breach. And the reason it's open is that the deepest quarter of the ocean has been visited by our instruments for barely one human lifetime, a place larger than nations, sampled at scattered points of light in an area of darkness we haven't begun to exhaust. Both possible resolutions would be profound, and it's worth being precise about why. If the limit holds, if decades of future exploration keep finding the fish stopping at the line, then that number, 8,200 meters and change, becomes something close to unique in biology, a hard physical boundary of a whole vertebrate way of being alive, written in osmotic chemistry, drawn on the map of a planet, and legible from a single equation. We don't possess many facts like that about life. We do not possess, arguably, any other quite like it. And if the limit breaks, if some camera someday records an unmistakable fish shape cruising a trench floor at 9 or 10,000 meters, then on that day we'll learn that vertebrate chemistry contains a spare key nobody's found in the laboratory. Some way of defending folded proteins or managing inflowing water that a century of physiology missed. And every textbook chapter behind this story will grow a thrilling new final page. The beautiful thing, the thing that makes this a privileged moment to be paying attention, is that the cameras are down there now. The expeditions are running. The juvenile snailfish filmed in the Izu Ogasawara Trench in 2022 drifted past a camera that exists because a small community of researchers decided the bottom quarter of our own planet deserved to be looked at. And every year they lower their instruments past the line and wait in the dark to see what comes to the light. That patience is the oldest tradition in this whole story. Everything we've followed descended from somebody deciding to keep looking after the sensible conclusion was already in, from John Ross trusting a brittle star on a sounding line, to Pickard and Walsh falling 7 miles in a steel ball on the strength of a hunch about gasoline, to a doctoral student measuring the chemistry of 5 cold fish from a trap. If this journey down the water column gave you even one moment where the world felt bigger than it did before? The like button is the closest thing this channel has to a lander surfacing with good news. And subscribing means you'll be on deck for the next expedition because we have a lot of ocean left and no intention of staying in the shallows. And I do want to know, down in the comments, where you'd place your bet, and I mean an actual position, stated in meters or feet. Does the line hold forever, or does some soft, patient, faintly smiling fish eventually find the door? The scientists themselves are split on that question, so whatever you answer, you're in professional company. Argue it out below. The best exchanges have a way of steering where this channel points its cameras next. The bet itself, though, should be placed with clear eyes, and here's the ledger as it stands tonight. The deepest confirmed fish on Earth swam past a lens at 27,349 feet, inside the narrow band where a 30-year-old chemical hypothesis predicts its kind must stop. Below it lie another 8,500 feet of living, swarming, permanently unlit ocean in the Challenger Deep alone, in which no vertebrate has ever been documented. Whether that final country is closed by an unbreakable law of osmotic arithmetic or merely waiting out the slow approach of an evolutionary locksmith is a question no one alive can currently answer. The measurements that will decide it are being taken now, trench by trench, deployment by deployment, in the coldest, heaviest water on this world. Until they're in, the last mile and a half of our own planet belongs to the amphipods, the sea cucumbers, the giant single cells, and the microbes, and the honest edge of human knowledge sits at 8,336 meters, in the beam of a borrowed light, watching a small pink fish swim calmly along the border of the possible.